For decades, over 90% of all genetic research on Earth was built on the DNA of one group, Europeans. That means for millions of people around the world, modern medicine was designed without them in mind. Now, a massive new wave of research is finally changing that, comparing Korean and European genomes at a scale never attempted before. And what scientists are uncovering is reshaping the way we think about human genetics itself. To understand why this matters, you have to understand just how lopsided the world of genetic science has been. Imagine trying to build a map of the entire planet, but only ever surveying one continent. That is essentially what happened with human genetics for the better part of 2 decades. Massive research projects, biobanks holding the genetic information of hundreds of thousands of volunteers, were assembled almost entirely in Europe and North America. The UK Biobank alone holds deep genetic and health data on roughly 500,000 British citizens. Similar projects sprang up across Scandinavia and Iceland. These became the bedrock of modern genetic science, the reference points that doctors, researchers, and pharmaceutical companies use to understand which genes are linked to disease, which mutations raise your risk of diabetes or heart disease, and which drugs will actually work in your body. But here's the problem. Asia is home to roughly 59% of the entire human population. And yet, for years, only about 4% of participants in genome-wide association studies, the massive comparative studies that hunt for links between genes and traits, came from Asian populations. That is not a small gap. That is an entire hemisphere of humanity being left out of the blueprint. And when an entire hemisphere is left out, the consequences are not abstract. They show up in real hospitals, in real diagnoses, in real prescriptions that do not work the way they are supposed to. Before we go further, it helps to understand what scientists are actually doing when they say they're comparing 2 populations' DNA. They are not lining up 2 individual people and checking for differences letter by letter. Instead, they are comparing patterns across tens of thousands of genomes at once, looking for spots in the genetic code where a particular variant shows up more often in one population than another, and then checking whether that variant is statistically linked to a measurable trait, like blood pressure, cholesterol, or disease risk. It is a bit like comparing 2 enormous libraries, not by reading every single book cover to cover, but by tracking which specific words and phrases appear more frequently in one collection versus the other, and then figuring out what those patterns actually mean. This is where Korea enters the story. Korean researchers, frustrated by this imbalance, began building their own massive genetic resources, projects like the Korean Genome and Epidemiology Study and the Korean Cancer Prevention Study, biobanks stocked with the genetic and health data of well over 150,000 Korean citizens. The goal was not just to catch up to European research. The goal was to finally ask a deeper question. What do human genomes actually look like when you compare a population that evolved and migrated across the plains and river valleys of the Korean Peninsula against a population that evolved and migrated across the very different landscapes of Europe? So scientists did exactly that. They took this enormous trove of Korean genetic data and combined it with data from other major East Asian biobanks, including Biobank Japan and the Taiwan Biobank. And then they set it directly against the data from the UK Biobank, one of the largest European genetic databases on the planet. This kind of study is called a meta-analysis, and think of it like combining dozens of individual maps of different neighborhoods into one enormous high-resolution map of an entire country. The more maps you combine, the more detail emerges, the more you start to see patterns that were invisible before. And the patterns that emerged were extraordinary. Researchers identified more than 300 brand new genetic locations, what scientists call loci, that had never before been linked to any human trait. These were regions of the genome tied to 36 different measurable characteristics, things like cholesterol levels, blood pressure, thyroid function, and body composition. But it did not stop there. When the Korean data was combined with the other East Asian and European datasets in that massive meta-analysis, scientists uncovered more than 4,500 additional genetic locations that had not shown up as significant in any single study on its own. To put that scale in perspective, imagine 2 separate teams of surveyors mapping the same mountain range, one team walking the eastern slopes and the other the western slopes. Individually, each team produces a solid, useful map of the terrain in front of them. But it is only when their 2 maps are laid on top of one another that hidden ridgelines, hidden valleys, and entire peaks that neither team could see on their own suddenly snap into focus. That is essentially what happened when Korean genetic data was layered onto the existing body of European genetic research. Think about that for a moment. These genetic signals were essentially invisible until Korean data was added to the mix. They were hiding in plain sight, waiting for a population with the right genetic variation to reveal them. One discovery in particular stands out, and it is a perfect example of why genetic diversity in research actually matters, not just academically, but for real human health. Buried within this research is a gene called ALDH2. If you have ever wondered why some people turn bright red and feel their heart race after just one sip of wine or beer, while others can drink for hours without so much as a flush, the answer very often lies in this exact gene. ALDH2 controls an enzyme responsible for breaking down a toxic byproduct of alcohol metabolism called acetaldehyde. In people carrying a particular variant of this gene, acetaldehyde builds up rapidly in the bloodstream, causing that telltale flushing, along with nausea and a racing pulse. This variant is remarkably common across East Asian populations, including Koreans, but it is exceedingly rare among people of European ancestry. For a long time, this was viewed mostly as a curiosity, an interesting footnote about why certain people get the so-called Asian flush. But the deeper genetic research revealed something far more significant. That same ALDH2 variant does not just affect how you react to alcohol. Fine mapping studies, a technique that allows scientists to pinpoint the exact genetic variant most likely responsible for a biological effect, showed that this single mutation appears to be a likely causal factor across multiple different traits and health outcomes simultaneously. In other words, one tiny change in one gene is quietly influencing multiple systems in the body at once. And it is a change that is common in Korean and other East Asian populations, but almost absent in European ones. If researchers had only ever studied European genomes, this connection might never have been found at all, and that has real consequences, because ALDH2 variants have been linked in various studies to differences in cardiovascular risk and even certain cancer risks tied to alcohol exposure. Without Korean genetic data, an entire piece of the human health puzzle would still be missing. ALDH2 is not the only example of a gene where East Asian and European populations diverge in medically important ways. Another well-studied case involves a gene called CYP2C19, which plays a central role in how the liver metabolizes certain medications, including a widely prescribed blood thinner called clopidogrel, often used after heart attacks or stent placements to prevent dangerous blood clots. A variant of CYP2C19 that reduces the drug's effectiveness is significantly more common in East Asian populations, including Koreans, than in European populations. That means a standard dose that works perfectly well for the average European patient might be far less effective for a meaningful percentage of Korean patients, potentially leaving them at higher risk of a repeat cardiac event unless doctors know to check for this genetic variant and adjust treatment accordingly. This is precisely the kind of life or death detail that population-specific genetic research can uncover, and precisely the kind of detail that gets missed entirely when research is not diversified. Alcohol-related genetics tell a similarly layered story. Beyond the well-known flushing response, the ALDH2 variant, common in Korean and other East Asian populations, has also been studied in connection with esophageal cancer risk. Because the variant causes toxic acetaldehyde to build up in the body rather than being broken down efficiently, chronic alcohol consumption in people carrying this variant has been associated in multiple studies with a substantially elevated risk of certain cancers of the digestive tract. In European populations, where this variant is exceedingly rare, that particular risk pathway barely registers as a public health concern. But in Korea and across much of East Asia, it represents a genuinely significant piece of public health guidance, one that simply would not exist without population-specific genetic research. But perhaps the most surprising finding in all of this research is not about the differences between Koreans and Europeans. It is about how similar the two populations turn out to be in ways that matter most. Scientists have long studied something called heritability, which is essentially a measure of how much of the variation in a trait, like height or body weight, can be explained by genetics rather than by environment, diet, or lifestyle. When researchers calculated the heritability of height in Korean populations, they found it to be remarkably high, and strikingly almost identical to the heritability estimates calculated in European populations. The same held true for body mass index. Statistically, there was no significant difference between the two groups. Your height is shaped by genetics to almost exactly the same degree, whether your ancestors came from the Korean peninsula or from the hills of England. To arrive at these heritability numbers, researchers typically rely on massive statistical comparisons across large populations, examining how much of the variation in a trait tracks along with shared genetic variants versus how much appears to be shaped by environment, nutrition, and lifestyle. It is a completely different method from the classic twin studies you may have heard of, where scientists compare identical twins, who share virtually all of their DNA, against fraternal twins, who share only about half. Both approaches, the twin study method and the large-scale genomic method used in this Korean-European comparison, have independently arrived at strikingly similar conclusions over the years, which gives scientists real confidence that these heritability estimates reflect something genuinely consistent about human biology, rather than a statistical fluke tied to one particular dataset or population. This might not sound shocking on the surface, but it actually cuts against a very old and very persistent myth: the idea that different ethnic populations are somehow fundamentally different in how their biology works at the deepest level. What this research shows instead is something far more unifying. The underlying genetic architecture, the basic machinery by which genes influence traits like height and weight, functions in remarkably similar ways across human populations, even populations that have been geographically and reproductively separated for tens of thousands of years. The differences that do exist tend to be about which specific genetic variants are common or rare in a given population, not about some deeper, more fundamental biological divide. To really appreciate why Korean and European DNA look the way they do today, you have to rewind the clock way back tens of thousands of years to the earliest human migrations out of Africa. As early humans spread across the globe, small groups broke off, isolated by mountains, oceans, deserts, and sheer distance. Over thousands of generations, genetic mutations accumulated differently in each of these isolated groups, some through pure random chance, others because certain mutations offered a survival advantage in a specific environment, whether that meant surviving colder winters, resisting a local disease, or metabolizing a regional diet. The population that would eventually become Korean people has deep roots, tracing back to a blend of prehistoric groups from what is now China, Manchuria, and the Korean peninsula itself, groups that came together and mixed extensively during the Neolithic period and the Korean Bronze Age. Genetic studies of ancient DNA from cultures like the Hongshan culture in Manchuria have revealed Y chromosome lineages, the genetic markers passed down through the paternal line, that represent over half of the paternal lineages found in Korean men today. This tells us that the foundations of the modern Korean gene pool were being laid thousands of years before recorded history even began. Maternal genetic lineages tell a complementary part of this story. Mitochondrial DNA, which is passed down almost exclusively from mother to child, reveals a distinct set of haplogroups common among modern Koreans, including lineages known as D4, D4A, D4B, and several others, such as M7, M8, and B. The D4 lineage in particular stands out as the single most frequent maternal haplogroup found in Korean populations today, appearing at similar frequencies across different regions and even across different health study cohorts, suggesting a deep, stable maternal lineage stretching back many thousands of years. These mitochondrial patterns overlap significantly with lineages found in neighboring Chinese and Japanese populations, reflecting the shared deep ancestry of East Asian peoples while remaining clearly distinct from the maternal lineages that dominate in Europe. Interestingly, genetic research has also uncovered faint traces of what researchers describe as ancient contact with Indo-European populations, the broad ancestral group that eventually gave rise to most European populations. Around 3,500 years ago, waves of technology, including bronze working, horseback riding, and chariot warfare, spread from Indo-European groups into Mongolia, and from there filtered into the Korean peninsula. Some Y chromosome studies have even detected small traces of what appear to be European-associated paternal lineages within the modern Korean population. Though these lineages make up less than 1% of the total, it is a small, faint genetic echo, but it is a real one, a reminder that human populations have never been as isolated from one another as we sometimes imagine, even across staggering distances of geography and time. Meanwhile, the ancestors who eventually became European populations were undergoing their own complex journey. Multiple waves of migration swept across the European continent over thousands of years, from the earliest hunter-gatherers who arrived after the last ice age, to Neolithic farmers who migrated up from Anatolia, bringing agriculture with them, to later Bronze Age pastoralist groups sweeping in from the Eurasian steppe. Each wave left its own genetic fingerprint, layered on top of the last, eventually producing the complex genetic mosaic we now think of as European ancestry. Certain genetic markers, like specific mitochondrial DNA lineages and Y chromosome haplogroups, became especially common in different regions of Europe, tracing back to these distinct ancient migration events. Haplogroups like R1b and R1a, for instance, dominate the paternal lineages of Western and Northeastern Europe respectively, while a maternal lineage known as haplogroup H became, by a wide margin, the single most common mitochondrial lineage across the entire European continent. None of these lineages appear in any significant frequency in Korean populations, and the Korean lineages we just discussed appear only faintly, if at all, in Europe, underscoring just how thoroughly these two branches of the human family tree diverged after their ancestors went their separate ways tens of thousands of years ago. So when modern scientists line up a Korean genome next to a European genome, what they are really looking at is the accumulated genetic residue of two profoundly different but equally complex journeys across deep human history, different environments, different migrations, different periods of isolation and mixing. And yet, remarkably, underneath all of that divergence, The fundamental genetic blueprint, the basic rules by which genes build a human body, remain overwhelmingly the same. This brings us to why all of this actually matters beyond pure scientific curiosity, because the implications reach directly into the doctor's office and the pharmacy counter. One of the most powerful tools emerging from modern genetics is something called a polygenic risk score. Instead of looking at a single gene, scientists can now combine information from thousands of tiny genetic variations scattered across a person's entire genome to estimate their inherited risk for conditions like heart disease, type 2 diabetes, or certain cancers. These risk scores are becoming an increasingly important part of preventative medicine, helping doctors identify which patients might benefit most from early screening or lifestyle intervention long before symptoms ever appear. But here is the catch. A polygenic risk score is only as accurate as the data it was built on. If a risk score was calculated entirely using genetic data from European populations and then applied to someone of Korean or other East Asian ancestry, its accuracy can drop significantly. The genetic variants that predict risk in one population are not always the same variants, or do not carry the same weight in another population. This is not a hypothetical concern. Multiple studies have shown polygenic risk scores built entirely from European genetic data lose a substantial amount of their predictive power when applied to East Asian populations, sometimes explaining only a fraction of the risk variation they were able to explain in the original European cohort. It is a documented problem that researchers have been racing to solve, because a risk score that fails to work accurately outside of European populations is not just a scientific shortcoming, it is a direct driver of health inequality. It means that as personalized, genetically informed medicine becomes more common, an entire segment of the global population risks being left behind, receiving less accurate risk predictions, less effective preventative care, and by extension, worse health outcomes, simply because the underlying research never included people who looked like them. This is exactly the gap that the new wave of Korean genetic research is working to close. By building enormous, detailed genetic data sets from Korean and other East Asian populations, and then directly comparing and combining them with European data sets, researchers are not just cataloging curious differences between groups. They're actively rebuilding the foundation of genetic medicine to make it more accurate, more equitable, and more useful for everyone, regardless of ancestry. Every new locus identified, every variant like ALDH2 uncovered, every heritability estimate calculated adds another brick to a more complete and more just picture of human biology. It is also worth pausing here to address something important, because studies comparing DNA across ethnic groups can sometimes be misunderstood, or worse, misused, to suggest that different populations are fundamentally, hierarchically different from one another. The actual science says the opposite. Modern population genetics has consistently shown that the vast majority of human genetic variation, somewhere around 85 to 90% depending on the specific study, exists within populations, not between them. In other words, there's more genetic diversity among individual Koreans than there is, on average, between the broad genetic profile of Koreans and Europeans as groups. Human genetic variation does not sort itself into neat, discrete boxes that map cleanly onto the old, socially constructed idea of race. Instead, it flows in gradients shaped by geography, migration, and history, with populations blending gradually into one another rather than splitting into hard biological categories. What studies like this Korean-European comparison actually reveal is not a story of division, but a story of shared humanity expressed through countless small variations that, when studied properly and inclusively, can help every single person receive better, more personalized healthcare. It is worth remembering that a Korean person and a European person share, on average, more than 9% of their DNA in common, the same figure that holds true for any two humans on the planet, regardless of where their ancestors happen to live. The differences scientists are hunting for in studies like this one are not differences that separate humanity into distinct types. They are differences measured in fractions of a percent, subtle variations layered on top of an overwhelmingly shared genetic foundation, and it is precisely because that foundation is shared that discoveries made in one population, like the ALDH2 findings, can end up teaching us something valuable about human biology as a whole. Looking ahead, this research is only accelerating. Korean biobanks continue to grow, and scientists are working to expand genetic sampling even further, including efforts to eventually incorporate genetic data from North Korea, a population that has remained almost entirely absent from global genetic research due to decades of political isolation. Researchers are also pushing to include more diverse populations from across South and Southeast Asia, Africa, and Latin America, regions that remain dramatically underrepresented compared to their share of the global population. The ultimate goal is a genetic map of humanity that finally reflects the true, staggering diversity of our species, rather than one built almost entirely around a single continent's population. There is also a technological wave building right behind this research that will only make it more powerful. As gene editing tools and increasingly sophisticated genetic therapies move from experimental labs into real clinical treatments, the underlying genetic reference data those tools rely on becomes even more critical. A gene editing approach designed to correct a disease causing mutation needs to know, with precision, which version of a gene is actually present in a given patient's population, and whether editing it might interact with other nearby variants common in that group. Without broad, diverse genetic data sets like the ones now being built in Korea, these next-generation treatments risk repeating the same European-centered blind spot that has shaped genetic medicine for the past 20 years, just with far higher stakes attached. For everyday people, the impact of this research will likely be felt less as a single dramatic headline and more as a slow, steady improvement running quietly in the background of modern medicine. It will show up in more accurate genetic tests, in drug treatments calibrated to work effectively across different populations, in disease risk calculators that give Korean patients, and eventually patients from every background, numbers they can actually trust. It will show up in scientists finally being able to answer questions about human health that simply could not be answered when the data was drawn from only one corner of the world. And it will show up in something even harder to quantify, but no less real, a growing sense of trust, as more and more communities around the world can look at the foundations of modern medicine and see themselves genuinely represented within it, rather than treated as an afterthought, bolted onto a system built for someone else. Consider, too, what this means for something as simple as a routine medical checkup. A doctor calculating a patient's 10-year risk of heart disease is often relying, whether they realize it or not, on risk models built primarily from European genetic and health data. For a Korean patient, or any patient from an underrepresented population, that risk calculation may quietly be less accurate, nudging a screening decision, a medication dosage, or a preventative recommendation in a direction that does not truly fit their biology. Multiply that single small inaccuracy across millions of patients and thousands of clinical decisions made every single day, and you begin to see why closing this genetic research gap is not a minor academic footnote. It is one of the most consequential public health projects of our time, unfolding quietly in research labs rather than on the evening news. So when you hear about scientists comparing Korean DNA to European DNA, the real story is not about shocking hidden differences designed to divide one population from another. The real story is about a scientific community correcting a decades-old blind spot, and in doing so, uncovering genetic variants that had been hiding in plain sight, confirming just how similar humans really are beneath the surface, and building the foundation for a future where medicine finally works for everyone, not just the population that happened to be studied first. That is not just a shocking result. That is a genuinely important one, and it is a story that is still being written, one genome, one discovery, and one closed gap at a time. And the next chapter of that story is likely to include populations we have barely begun to study at all, meaning the most important discoveries in human genetics may still be waiting, quietly, just ahead of us.